Hello, I'm Philip Morris, and I'm joined tonight by David Havz
Hello.
to discuss the latest in science news and skepticism. These conversations used to happen over a drink in our local pub. The idea came about to bring these musings to the world of the tinterweb in the form of a podcast, and of course, we're still accompanied by a drink or two. Welcome to a Skeptics Night In. How are you, Havz?
Yeah, good mate. How are you?
Good. It's a shame we don't have Dave here today.
I know it is a shame, but he wants to go travelling, and he hasn't got the time to commit, basically.
That's fair enough.
Yeah.
It takes a lot of work to make this podcast happen. So
Yeah, it's true. And plus, we'll get guests on, and I'm sure Dave will come on as a guest in the future.
Yeah, definitely. We've got a couple of guests lined up for the next few episodes, and we'll hopefully get Dave on again.
Look forward to it.
So, what are you drinking, House?
I'm drinking Red Stripe tonight, which is a Jamaican lager, and on the can it says Jamaica Lager Beer, and it's 4.7%. I like
a bit lower than last week.
Yeah, a bit lower, a bit lower than the 5.6.
5.6. Yeah.
Yeah. What are you drinking, Phil?
I am drinking Old Moot cider.
As in Moot Point?
Well, I don't know. It says on the bottle, pronounced incorrectly since 1947, which is interesting because until I read that on the side of the bottle, I literally thought it was Old Mount cider. I completely didn't even notice that there wasn't an N in Moot. Yeah, I'm drinking the kiwi and lime flavour. It's a cider company established in Nelson, New Zealand. So we're very international tonight.
Yeah, I didn't have you as a fruit cider drinker, Phil.
Oh, it's summer, isn't it?
Yeah, I was looking for crabbies, but unfortunately the shop didn't have it, so I just stuck to lager.
Okay, so we're going to start the news section by talking about the recent awakening of the Philae lander.
Philae! Talk us...
Philae.
Philae, talk to us about Philae.
On Saturday the 13th of June, the hibernating lander Philae on Comet 67P made an 85-second transmission to the European Space Operations Centre in Darmstadt, Germany, transmitting over 300 packets of data. The European team still estimate a further 8,000 data packets being held on Philae's memory banks. But before we talk about that, let's just have a quick recap on the Rosetta mission. So the Philae lander was launched from the Rosetta spacecraft in October last year to land on the surface of the Comet 67P, except it didn't land down properly. Bounced twice, and the harpoons didn't manage to anchor the lander. Flew up into the air. 2 hours later, the lander came down to rest on an overhang. And what that meant was that the solar-powered batteries could charge. So the Philae lander was only operational for a little over 2 days, about 60 hours. And it spent that time collecting as much data as it could. And some of the data that it came back with was quite interesting. I've covered this in a recent blog post, so if you're interested in finding out some more details, go to that. But essentially what Philae has learned is that small comets like 67P have a very limited magnetic field, and it can be assumed that magnetism was not a force that governed the initial accumulation of material into the large terrestrial bodies that we see in the solar system today, like the Earth, the Moon. So there are other mechanisms that were more important to the aggregation of material in the solar system. What's next for Rosetta? Well, they wanted to drill into the surface of the comet, melt the material in a small oven that's on board Philae to analyse the results, but it might be that the lander doesn't actually have enough power to carry out that task. So what it will be doing is it will be taking temperature and magnetic field readings again, as well as more images. As the comet approaches the sun, there will be a significant warming on the surface, with the possibility of the comet breaking apart into two distinct bodies. The activity on the comet could be actually dangerous for Philae, but serendipitously, the final landing spot could provide it with the protection that it requires due to its sheltered position. So it's very hard to say what will happen over the next few months as the comet reaches its closest approach to the sun on the 13th of August of this year.
And the Rosetta team, they are quite hopeful, aren't they?
I mean, they have been hopeful for quite a few months. So they were expecting Philae to wake up in April, and it took them until June. So
yeah. But they did say that they think that Philae... Sorry, how's it pronounced?
Philae.
Sorry, Philae. It is Philae, isn't it? That it may have woken up before most recently.
Yes. Yeah. That's interesting. Yeah, they reckon that it was actually taking some measurements, but it can only transmit information to Rosetta during a 2-hour window when Rosetta's closest to the landing site. It hasn't actually relayed that information back, but they think it might have been collecting data. So that's, interesting. And there's a lot of data come back from the actual lander itself. Yeah. So
I mean, hopefully we'll get those further 8,000 data packets. Fingers crossed, Phil. Fingers crossed. So I'll be covering our second news story for tonight, and the news story is called "Organs on Chips."
Do we get gravy with it?
Doesn't that sound appealing? I think Harvard University would be quite angry if you put gravy on it. So essentially, at Harvard University, at the Weiss Institute, they made these silicon chips that mimic the function of living human organs. And it's actually won the Design of the Year award from the Design Museum in London. And so the scientists at Harvard University, they placed human cells from different tissues onto these chips to study how different organs work. They have a lung on a chip, and it contracts and relaxes as air passes over the cells like lungs would do naturally. And the Weiss Institute essentially says that the devices could be used as an alternative to animal testing for drug development, safety of cosmetics, or toxicity of chemicals.
So what are we talking about here? Are we talking about... So you're saying you literally just put a lung on top of the chip? It's software, is it?
Kind of. I mean, it's very complicated, as you can imagine. Basically, it's a clear silicon cube. Within it, you have different compartments, and these compartments contain the stuff that the cells need to use to live, and the different mechanisms to simulate the certain organ. And then you have these tubes running through it that contain the human cells of like lung tissue or heart tissue or intestinal tissue. Essentially, all these compartments together, they mimic the function of the specific organ. Yeah, I mean, it's a very complicated piece of kit. We'll put a link in the show notes for anyone who's interested to have a deeper look into it. But it offers these quite exciting alternatives to animal testing, basically. Because if we can, experiment without using animals, then we probably should.
What sort of things would they experiment?
Well, they want to build 10 of these different organs on chips, and then they want to link them all together to create this automated instrument to mimic whole body physiology.
Okay.
And the instrument will control like fluid flow, cell viability, and then because it's clear, it permits real-time observation of the tissues, and you can analyze the complex, biochemical functions. So before like human testing, they could test it on these kind of organ cultures or organ mimicry devices. I don't know what you want to call them.
So it essentially makes the whole thing's a lot more efficient.
Oh, it's a hell of a lot more efficient and a lot more cheap as well.
Cheaper as well.
Because most human trials cost a hell of a ton to even get to human testing.
Surely you'd still have to do
cheap alternative.
You'd have to do human trials as well, but presumably the drugs would be safer at that point anyway. By the time it gets to proper human
clinical
trials.
Studies can take years to complete and test a single compound, and can cost over $2 million. During that time, innumerable animal lives are lost, and the process often fails to predict human responses because traditional animal models don't necessarily mimic human physiology.
Yeah, because you hear a lot of studies that talk about, we tested this in rats and it came up with this idea, but then, a rat's physiology is slightly different to a person's physiology, so what's bad for a rat, not necessarily bad for a person.
Exactly. You have to take those studies with a massive grain of salt, which most, science journalists don't. So yeah, I think it could be an exciting new piece of technology, and I look forward to hearing more about them in the future.
We're going to be looking at Jurassic Park. Considering the new movie Jurassic World came out earlier this month, how possible is it to make an extinct animal in the lab? In the original movie, Jurassic Park created a genome of dinosaurs using the blood from mosquitoes trapped in fossilized tree sap called amber. The idea was that the mosquito would bite a dinosaur, land on the bark of a tree, and get trapped inside the sap, which would then fossilize and get dug up millions of years later. The DNA was then extracted from the abdomen of the insect, and scientists used frogs to fill in the gene sequence gaps. So we're going to be looking into the viability of creating extinct animals in the lab based on these techniques and possibly other techniques.
So as Phil said, in Jurassic Park, they create dinosaur genomes from within mosquitoes, and then they use like frog DNA to patch up the bits. Funnily enough, it's not far from reality. Well, in the case of dinosaurs, unfortunately, the amount of time that's passed has ruined our chances of reviving the T-Rex or the Velociraptor, as under ideal conditions, DNA can only last a few million years. Due to cosmic radiation, it basically cuts the DNA to bits and it's unusable.
I had an interesting stat here on the half-life of DNA. They worked it out about 521 years.
Is that within like normal room temperature?
Yeah, well, this is it. They estimated that even at the best conditions, where the samples are kept dry and cold to a temperature of about 23 degrees Fahrenheit, it would take only 6.8 million years for the genome to degrade completely.
Exactly. Dinosaurs like T-Rex lived more than 50 million years ago, so it's completely unviable to be able to bring them back. Unfortunately, well, or very fortunately,
depends on your perspective.
According to the lessons of Jurassic Park. So thanks to basically DNA decay, there just isn't enough genome left to attempt to revive them.
Yeah. Also, I was reading an article about this fella Jack Horner, who has written a book, How to Build a Dinosaur. Now, I'm giving you a plug. I don't know why, but here we go. Free. You can have that. Basically, what he's saying is that
you could probably download a torrent for it. You could get it for free online.
Yeah, you can get it for free online. Yeah.
There we go.
Just download it. Nice. He basically he says that you could probably reverse engineer a dinosaur from modern day birds. So you're only looking at theropod dinosaurs here, which are like those ones that walked on two legs, like T-Rex, Velociraptor. So what he did was he was looking at a developing chicken embryo, and looking at the tail, he expected to see about 4 1/8 vertebrae in the spine, but through his microscope he actually picked out 16 vertebrae, which was effectively like a reptilian tail. But what happened was, as the bird developed in the egg, the tail became shorter and shorter, and when it finally hatched, it only had about 5 vertebrae.
Ah, right, so they've like fused together.
Yeah. So he was saying that the blueprint for a dinosaur is still in birds. If you engineer the birds correctly during development, you could actually create these traits from a dinosaur rather than the evolved traits that they've now got from a bird. So, for example, the claws show resemblance to the wings of a modern day bird. Teeth, chicken's teeth, hen's teeth.
They've done that. They've done it.
They've done it already.
They've given chickens teeth or teeth-like structures.
Amazing.
Yeah, it's pretty cool.
That is a pretty rare find.
Yeah,
rare as hen's teeth.
I think they. Very good, very good. Yeah, so I think what they did there, they like knocked out a certain gene, and then this chicken just developed teeth.
What he does say though is that it would take within 100 to 150 years to actually be able to do this, given the understanding of genetics we have at the moment.
Oh yeah, it's
because
I mean,
the genome's
to me
so
it sounds
huge.
completely unviable. But what about other animals like the woolly mammoth? Because woolly mammoths were alive only about 4,000 years ago and lived in cold climates that tended to freeze the soft tissue once they've died. So as a result, we have a lot of mammoth mummies with access to pristine DNA. So is anyone trying to do this? Is anyone trying to bring back the woolly mammoth?
Yes.
Well, there's a yes. Essentially. Well done, Phil. You win
Nice.
today. Yeah. So there's a controversial South Korean geneticist called Hwang Woo-suk. I think that's how you pronounce it. I hope that's how you pronounce it. I'm sorry if I offended any South Koreans. So basically, he's hoping to take the traditional approach of cloning techniques and outlined in Jurassic Park he's hoping to discover a cell that's well preserved enough to contain a complete intact copy of the mammoth genome, which can then be implanted into an elephant egg to create a clone, very similar to the method used to clone Dolly the sheep. However, so far the mammoth DNA being found has been shattered into chunks that are only around a couple of hundred of base pairs long, and also they can be contaminated by the genomes of mitochondria within the cells and the bacteria that were there when the mammoth died, and also any random flecks of organic matter that managed to make its way into the sample. So, it is possible to reconstruct a mammoth's genome, out of this genetic soup, but it's very difficult. So it's essentially a jigsaw with 17 million pieces and no corners.
Oh.
But scientists are getting close. They're using computers to basically solve the problem for them, because computers are a lot better at these sort of puzzles than us. But that genome they create isn't complete, because it's missing repetitive sections, because these pieces are very hard to put together, and any chunks that didn't survive. So if you lost something, you're obviously not going to be able to get that back into the genome sequence.
So, like they did with the DNA of a frog from Jurassic Park, could they do the same thing with an elephant, just stick in elephant DNA in those gaps that they've got? Is it a bit more complicated than that?
Exactly it.
Nice.
So you could compare the DNA against the Asian elephant, because that's the closest living relative to the woolly mammoth, and use those differences to construct a hybrid pachyderm. And exactly as you said, it's similar how to the frog DNA was used to patch up the holes in the dinosaur genome. They'd use a similar system, so it's certainly possible. And even there's actually been research done where genes from a woolly mammoth were inserted into the African elephant to give them cold resistance.
What? So you can make an African elephant with just loads of hair?
Yeah,
like a mullet.
Well, maybe not hair, but I don't know what they
dreadlocks.
But all
actually look
all
like
I know
the
is
elephants off the Jungle Book.
Oh my god, that'd be amazing. Essentially, you'd be creating that, yeah.
Yeah.
Well, they didn't just do this so they could give an elephant dreads. The researchers hoped that this would allow Asian elephants to migrate from its shrinking ancestral habitat in order to save the species from extinction. So they'd insert these cold-resistant genes into these Asian elephants so that they could live further up north, thus saving their species' life. So basically, yeah, we're probably a fair way off yet, but we're getting certainly real close. I'd love to clone a mammoth.
Yeah, that'd be awesome. I'd definitely go to that zoo.
Yeah, definitely. We should just set them free in Siberia or something. Russians might not be happy with it. They're not even up for gay marriage, let alone, making a mammoth. God. Maybe Canada. North of Canada. There's no one there.
It might give polar bears something to eat. Maybe a bit smaller.
Yeah, that's a good point. We'd have to
solve that problem.
Problem solved. Did polar bears live south enough for that?
No, not really. No, they're more sort of sea ice area, whereas mammoths were steppes, cold tundra. Because obviously they couldn't live in Arctic desert because there's nothing to eat.
Yeah, exactly. There's nothing there. China? You could put them in China. Oh no, the Chinese would love to eat that. Like woolly mammoth tusk could be their new cure-all. Mammoth tusk cures cancer.
Mammoth tusk cures cancer. I like it.
You know what China's like?
Yeah.
Everything's a cure there.
You can 3D print them now anyway, so it's fine.
Yeah, that was an amazing story. The rhino horn, wasn't it? Rhino horn? They're going to 3D print loads of rhino horns.
Yeah, and then basically just flood the market. Because it's sort of it's almost identical to a rhino horn.
Yeah, and they're basically saying they're going to implant DNA in it, so to fool tests.
Yeah, it would just sort of ruin the market for it and stop people hunting.
It's basically literally like China that are killing most of the world's glamorized rare animals.
Yeah, they're
like the
on
whales,
purpose.
The rhinos. Yeah, on purpose. That's a good point to make. Yeah, we're all killing endangered animals by living how we do, but they're actually purposefully doing it.
Yeah.
So now we're going to play the game Seeing Through the Myths. And normally this is played with Dave and Phil, but today it's just me and Phil, so we'll be doing things a little bit differently today. So I have 4 statements, and some of them are true, some of them are commonly held beliefs or myths. And I'm going to go through them individually, and Phil has to guess which is a myth and which is a truth. Right, so number 1, blood is blue.
Oh, yeah, okay, I've definitely heard this one. Blood is blue until it's oxygenated, which makes it red.
Exactly it. Yeah.
But it's not true. I read somewhere that it's a myth. I'm pretty sure that one's a myth.
It's a myth.
Yay!
Yeah, so they're not quite sure where this one came from. They think it came from old textbook diagrams which showed blood as being blue. But
I think when you look at when you look at veins in your arms, they look blue,
don't they?
But aren't they actually arteries? The ones that you can see, or is
No,
that
the ones you can see are veins because
okay? Yeah,
because
yeah, but
veins
it looks blue anyway.
they're powered by like muscular action. So they normally go towards the surface so that
Oh
you
right.
can pump blood through them using the movement of your arms and legs. And then
Oh right, that's clever. I didn't
arterial
know
is
that.
pumped by the heart more than anything. So oxygen-rich blood, like arterial blood, is bright red, and then carbon dioxide-rich blood, venous blood, is darker red. So number 2, cows have 800 different blood types.
What? Humans have 4, cats have 9 lives. 800.
But
How many?
not 9 blood
800
types.
different.
Yeah, 800.
Why would they need 800 types of blood? Could it be from all the It happens because of all the alien experiments that have been done on them over the years.
Obviously, yeah.
Yeah, the UFO experiments. I don't know about this one. I don't know why they'd need 800. Why would cows have?
Blood
I don't. I just don't buy this one. 800 seems like too much. Too much of a leap.
What are you going for?
Although I've never heard of this one, so it could be true. No, I think it's a myth. I don't know. I don't believe that one.
This one is true.
Oh, Havz, damn it.
So cats have 9 lives, but they have 11 blood types.
11 blood types. Crikey.
Yeah, and dogs have 4.
Cows have 800 of those.
Yeah, that's amazing, isn't it?
There's not even 800 letters.
I wonder how they do it.
Oh,
You'll just
mental.
have number 1, 2.
Yeah.
But yeah, I thought that was incredible. I mean,
That's amazing.
that
yeah.
sounds wrong to me, but this did come from a fairly credible site. Yeah, so basically, I want to talk a bit more about blood types. So basically, blood types come from the receptors on the blood cells. If you have blood type A, you have receptor type A. And if you have B, you have receptor type B. AB, you have both. And then O is none of them. And that's why it's a universal donator, but not a universal receiver, because AB is a universal receiver. And I'm not going to talk too much about it, but on the show notes, we're going to put up a link to a podcast by How Stuff Works, because they've got an amazing short podcast on blood types, and it's really, really interesting. So number 3, the average adult has 14 to 18 pints of blood.
14 to 18.
Yeah, about as much as you drink on the weekend, Phil.
14 to 18 pints of blood. Yeah, I guess so. I mean, yeah. I don't see why that wouldn't be true. I mean, you've got a lot of small capillaries and things like that, and that's just got to mount up. So true? I think it's true. Yeah.
That one is true. Yeah, essentially, I put this one in because I came across it today, and I always thought it was less. I always thought it was about 8 pints, just from remembering from school, like primary school. But yeah, no, I read that today, and that surprised me, so I wanted to put that in. But well done, you got that one.
Cool.
Okay, so the final one. You cannot donate blood if you're gay.
What? I think we're progressive enough to have gotten rid of that one by now. If that used to be the case, it may have been the case before, but I mean, they wouldn't knowingly inject somebody with. Surely they test the blood to make sure that there's no diseases in it. I don't know. I don't know.
What are you going for, Phil?
I don't think it's true. I can't see how in the modern society that can be true.
Oh, I thought I was going to catch you out on this one. Yeah, this one is a myth. Yeah, but it's not that simple. So,
okay.
Gay men can give blood, but only if you've been celibate for 12 months.
Really?
Yeah, because essentially, I think where this comes from is that the window to detect HIV can be as short as 7 to 21 days using RNA testing. But I think the most commonly used method, I think the most accurate method is something called serology testing. They can detect HIV at around 3 months. So this is why if you go in to get tested for HIV, you have to go in like 3 months later for another test because the antibodies can hide within the cells. And 3% of the population will still not test positive after 3 months. I think the reason why they've set the bar quite high is gay men are a high-risk group of people for HIV.
Well, that's just statistically we're talking now, I suppose.
Yeah, statistically, yeah. They don't accept blood from sex workers. You can't donate blood within 12 months of having sex with a sex worker. You can't give blood if you've been to North Africa within 12 months. So there's lots of these regulations because they don't want to give blood to someone and then they find out they're HIV positive later down the road. So they're just limiting their risks. I mean, there is a lot of controversy surrounding it. There are a lot of people saying this is rooted in homophobia rather than protection. I mean, I think they could.
I think you have to look at
they could probably shorten it to maybe 6 months. But I think always you should play safe rather than sorry.
It's based on statistics and
exactly. Yeah,
safety.
It's all about the numbers. I don't personally. I don't think there are any political influences.
No,
because otherwise they just wouldn't allow it at all.
Oh, good job, Havz. What'd I get there? 3 out of 4.
Yeah, you got 3 out of 4. Good job.
That's pretty cool. Oh, I'm pretty chuffed with that. Yeah.
I thought I was going to get you with the last one.
Yeah, I nearly went with It's True, but
because no, I've heard about that myth before that you just can't donate blood. Period.
I hope you can't donate that.
You definitely can't donate that stuff. Oh no! Sorry, women of the world.
Bringing the tone down here. So moving on to our main topic today, which is linked with the subject of the current Longitude Prize 2014. The Longitude Prize was a prize offered by the British government in 1714 for a simple method of determining a ship's longitude at sea. The prize was awarded to John Harrison, a carpenter and clockmaker who built a watch that was accurate at sea. The clock, which was the timepiece which made Rodney and Del Boy rich in the classic sitcom Only Fools and Horses, was revolutionary for being able to work on a constantly moving ship and was resistant to changes in air temperature and humidity. The idea was simple. If you could accurately keep Greenwich Mean Time, you could accurately estimate your longitude by comparing Greenwich Mean Time to the local time, which can be determined by astrological observations. So we're talking stars or the sun at its zenith. He eventually received £23,000 for solving the longitude problem and saved countless lives from the dangers of shipwrecks caused by poor navigation. Now, 300 years later, the British government has revived the Longitude Prize, but this time opened the topic out to a public vote. 6 topics were shortlisted. Flight, how can we fly without damaging the environment? The second one was food, how can we ensure everyone has nutritious, sustainable food? Thirdly, antibiotics, how can we prevent the rise of resistance to antibiotics? Fourthly, paralysis, how can we restore movement to people with paralysis? Fifth, water, how can we ensure everyone has access to safe and secure drinking water? And finally, the 6th one, dementia, how can we help people with dementia live independently for longer? This was put to a vote, and in May 2014, the results were out, and the public had voted to choose antibiotics as the focus for the Longitude Prize 2014. So we are going to be discussing antibiotic resistance. What is it? How bad is it? And what sort of things are we attempting to do to solve the problem?
Yeah, I think the public chose wisely there. I mean, obviously they're all very, very serious issues.
Yeah. I mean, you got your food and water and stuff, fine. But
I mean,
that's okay. But I mean, if 40% of the population are dying from antibiotic resistant bacteria,
exactly,
I mean, food and water are the least of your worries.
Problem with food and water,
Yeah.
I mean, if a fifth of the population died, then we wouldn't have so many problems with food and water. So I think it was a good decision there.
Yeah, definitely. I mean, even with the others as well. I mean, paralysis, dementia, and flight, all of them are pointless without.
Yeah. I mean, dementia is and it's going to keep going on the rise because people are getting older and older.
Yeah, but they won't be if all the strains of bacteria are resistant to our antibiotics.
Well, that's a very good
life
point,
life expectancy will reduce. Dementia won't be a problem.
Significantly. Yeah, exactly.
So yeah, overall,
it's
definitely
the most
important
issue there, I think.
Yeah, definitely, I agree.
So yeah, this is an actual real problem. Is really like most people will only come across the problem when they go to their GP and their GP won't give them an antibiotic. And they go, that's bloody ridiculous. Why am I paying taxpayers' dollars for,
Yeah, for things like, I've got the flu, give me some antibiotics. The flu's a virus, so antibiotics won't work.
Yeah.
So, but I think GPs sometimes will prescribe antibiotics to problem patients who are demanding it when they've got things like a cold or a flu or something. just
Yeah.
to keep them quiet, which is of
I
course
mean,
bad practice. But
I think we're very good nowadays. I mean, I remember being like a child, and I don't know whether things are different with children. I don't know whether GPs are a lot more giving with children, because I remember I'd get given antibiotics all the time as a child.
yeah,
Like, I'd go in with, like, I was ill, and they'd be like, it's probably viral, but take these antibiotics just in case. And, like, I don't know whether that was because we weren't so concerned about antibiotic resistance, like, 15 years ago, or whether they're just a lot more, a lot safer when they're dealing with children.
I mean,
But
I had
obviously, like,
I remember. Well, I was very small. I was only about 4, and for some reason I was ill. I was given amoxicillin, and I had an anaphylactic reaction to it. So I actually can't. I can't have penicillin-based antibiotics.
okay.
If I go to the doctors and they have to prescribe an antibiotic to me, they have to give me erythromycin, which isn't based in
Okay.
penicillin.
Yeah,
It's not an
yeah.
alien. It's not an alien.
Isn't that like the last kind of the last thing they give you before it's over, basically? Like the last solution. If antibiotics don't work, they just give you that.
Yeah, I don't know. Maybe. I hope not. If I
No,
could
to be
only
fair,
have the last resort,
well, no, but they're the only kind of antibiotics that actually work. Antibiotic resistance is a serious problem, and in the USA, the CDC, the Center for Disease Control and Prevention, they state that 23,000 people die every year from drug-resistant bacteria, and that's just in the USA alone. And many more die from conditions complicated by these drug-resistant bacteria. That's quite a large number, isn't it? For
yeah,
just the USA alone, that's maybe what, 1% of the population?
yeah. I
And,
mean,
these people would have lived, like, I don't know, maybe 25 years ago. They like, all of those people would be alive now, but it's just because of the drug resistance
yeah. I mean,
that's been developed by these bacteria.
this sort of figures pre-penicillin would have been about 40% mortality
What, bacteria,
from bacterial
infections?
infections. Yeah,
40%?
I think it was 40%
No,
mortality.
surely not, because
But if you
surely,
think
like,
about
I've,
all of
I've
the
had bacterial infections before and probably haven't been given antibiotics and I've been fine.
yeah, but we're talking about like serious ones.
Okay, okay.
Tuberculosis to begin
Oh,
with.
that was a major killer, yeah.
Yeah, you've got any sort of gangrene or anything like that. You'd have to have amputations for
Oh,
those.
yeah, of course, yeah.
There's all sorts of really bad bacterial infections out there that kill that we can cure with antibiotics now.
Barely. I mean, it's changing. Well, the FDA, they've only approved 2 new classes of antibiotics since 1998, 5 new classes of antibiotics in the last 45 years. So the majority of the antibiotics we use today were developed before 1968. The main ones we have, like with penicillin, we just can't replicate that sort of efficiency. Like, I think one of the major drug companies put $1 billion into creating a new class of antibiotics, and
Is
what they
this
ended up creating,
GS? Is it GSX? Is it?
GlaxoSmithKline.
Yeah, that's it. GSK.
Yeah, it's
Yeah.
probably GlaxoSmithKline. Yeah, they put $1 billion into creating a new class of antibiotics, and what ended up happening was this new class they created, it became drug resistant to it in 2 weeks.
2 weeks.
So I'm pretty sure it was 2 weeks. I'm a bit hazy on that part, but
Wow.
it wasn't very long at all.
So it's
And then
just
they
a
just
it's
kind of
just a lack
thought,
of
well,
efficiency for new antibiotics then.
they're just not finding new classes, so gonna need to take a different route, because currently there doesn't seem to be another way around it.
Yeah, I mean it's not like you
'Cause-
can stop bacteria from evolving because that's essentially what's happening here is bacteria evolve to be resistant to the toxins that you're giving to inhibit their growth, and they evolve a lot
Exactly, yeah.
faster than large multicellular organisms because they're
Oh
just a
yeah,
single cell
definitely.
and their life cycles are small.
I mean, I went
Yeah.
to my GP the other day with what I suspect is a chest infection or possibly bacterial pneumonia. He said it was probably a viral infection, but because I'm into science and he could tell that, he gave me a dose of a very strong dose of amoxicillin, so that if my condition didn't get better or if I started to produce certain symptoms, he said take the antibiotics then.
Right.
Eventually it happened and I had to take the antibiotics. what's interesting, because I researched amoxicillin after he gave it to me, and essentially most bacteria are resistant to it now.
Really?
And yeah, and he even said when he gave it to me, like, it probably won't work.
Is that why he had to give you in a strong dose as well?
Yeah, so I had to take 3 a day, and yeah, so I think they put in such strong doses to kind of counteract this resistance, or at least try and give it a good shot at killing the bacteria.
The sort of things we're talking about here, which kick off these bacterial resistances, is a lot of the time it's down to bad practice, and sometimes it's down to just misunderstanding by the public of what they should be doing and how antibiotics work. So
Definitely, yeah.
I spoke a bit about earlier GPs prescribing antibiotics for viral infections like the flu or cold or something to insistent patients who really don't understand the science. But you also get people who they'll be set on a course of antibiotics to clear up a bacterial infection, and when their symptoms get better, they stop taking the pills, they stop their course. Instead of doing the entire course of antibiotics to make sure that you've gotten rid of the infection, they stop when the symptoms disappear. And that hasn't gotten rid of the infection, and bacterial resistances can grow from that sort of thing as well. If you cut out that bad practice, it will slow antibiotic resistance down. So it's important, I think, that the public have an understanding of what the problem is and how we can all do something to slow it down. If we slow it down, we can find a solution faster.
Yeah, exactly, Phil. It's just like before, when I was saying I was ill, my housemate, she said, "Oh, do you want some amoxicillin?" Because she had some for a
Oh
previous
my god.
infection that she didn't take. She didn't end up
Yeah.
taking it in the end, and she said, "Oh, do you want mine?" And I was like, "Well, no, you shouldn't really do that. You shouldn't like save antibiotics, and you shouldn't give them to someone who hasn't been prescribed them. You always get them by prescription." And I think doctors doing a lot more now is being very careful with the antibiotics they give out, because they have to be, because it's getting pretty dire. I mean, there's resistance popping up all over the place. And one of the main, right, but there are two main reasons for this really. Is because when you're growing growing beef, raising cattle, they use a hell of a lot of antibiotics
Oh yeah, I've heard of
um,
this.
to raise them, so to improve their yields, basically, so
Yeah,
less
it's like a
cows
growth.
die.
How does that work? Is it like a growth accelerant or something? How does that affect?
I think it's two-part. I think it can affect growth hormones, which produce more meat, but
Right.
also it keeps cows from dying, basically, from the infections they would get from being in such close quarters with each other. Within the meat producing industry, they use around 300 different antibiotics, which is incredible. I mean, this is where we're getting these resistance problems with, because all, like a ton of the antibiotics that we could use are being used on our cows. The second problem is places like China and India. Anywhere really that the regulation of antibiotics either doesn't exist or no one cares. There was a Panorama documentary recently about antibiotic resistance, and they went over to, I think it was China,
It was
and
India. No, it was India.
or was it
They're in
India?
India. Yeah, I
Yeah,
saw it.
and they were buying strong antibiotics over the counter with no prescription, and you could even buy them in single doses. You could buy them in not the whole prescription, but a single dose,
Yeah, which is a
which is
terrible
crazy.
idea because it's not effective. A single dose of antibiotics is just not effective. You have to have a full
And like,
course.
and there are real problems happening over there right now, and it's a fascinating documentary. We'll publish it in the show notes.
Yeah, so what are some of the ideas that can help with this problem? I mean, how can we create more antibiotics?
So originally, antibiotics came about by Alexander Fleming. He discovered penicillin, and what he discovered was within these bacterial cultures, there were these rings around these bacterial cultures where nothing grew. And what he discovered was penicillin. It was giving off these bacteria-killing products, and that's how penicillin was invented, and it saved countless lives. I don't even want to know how many. It's a huge number of lives since. And most antibiotics work on the exact same principle as penicillin. And throughout the years, we found different types of methods of killing bacteria, but they're all centered around these couple of principles.
we're looking at trying to find bacterial cultures that we haven't had in the past, that we've been unable to culture in the past. It's difficult to culture some bacteria because they require specific nutrients and things that are specific to their environment. So say you took a sample of soil and you try to culture the bacteria from the soil in a lab. Only 1% of the bacteria that you culture from that soil will end up on the petri dish. The challenge here is to try and get the bacteria that you wouldn't get that dominate in the lab. So what they did is they grew the cultures in situ, and then once they'd managed to grow the cultures in the field, they were able to take them back and grow them further in the laboratory and see what the effects they have on other bacteria, whether they're able to produce toxins that are bacteria resistant. And what they found was they were able to identify approximately 50 different antibiotic drugs, essentially. And one of the most promising ones they found recently is called Teixobactin, and it's a brand new class, it was discovered in early 2015 and was published in the journal Nature. Essentially, it was grown using this new technique, and it was been shown to kill things like staph infections and tuberculosis. So this could be a really valuable antibiotic drug if it gets through all trials and everything. And this technique could find loads of new antibiotic classes, hopefully, but we have no way of knowing that just yet.
Well, what's really interesting, which I really hope becomes something, is that essentially insects do really well. Like they spread really fast, they're very genetically diverse, and a lot of the invasive species of insects, they travel a long way, like locusts, which means they are exposed to a lot of microbes. So researchers got thinking, like, if they're traveling so far, they're encountering all of these microbes, they must have really good immune systems and really good ways of dealing with bacterial infections. So yeah, in 2012, they decided to inspect these invasive insect species, which were hypothesized to have strong immune systems, and they were called the Harlequin ladybird beetles, and they successfully outcompeted native beetles the world over, so they're very successful. And what this guy discovered, Vilinskas, his name is Vilsinskas. I'm so sorry if I pronounced that wrong. I'm terrible. I shouldn't be allowed to read or write.
Every week.
So Vilinskas, he discovered more than 50 novel antimicrobial compounds in this one species of beetle. And that's not just antibacterial, that's antimicrobial. So that goes for fungi, viruses, and parasites. And one compound, which has been called harmonine after the Harlequin ladybird beetles, demonstrated activity against both tuberculosis and MRSA. The guy's name, his group of researchers is now making chemical modifications to harmonine to produce even more potent antibiotics. So it's really exciting, this piece of research, actually. I love it. Yeah, I love the idea
Cool.
that they saw that these insects are doing really well and they're
Yeah,
traveling
it's a
far
really
and
good
wide. They're
idea.
like,
It's an incredible idea. I mean, that
yeah, and they're like, wait a
it's
minute,
innovative.
like, how are they doing so well against all the bacteria and viruses and parasites that they'd come against? And I can't believe they found so many, 50 in one species of beetle. We don't really want to throw all this information at you because there's just so much of it, and I feel like we've gone too far already. We'll put some links up because it's amazing what
Yeah, there's a mine of
science is
information
doing
out
for the
there.
world.
Yeah.
There's a lot being done. I mean, it's a huge problem. A lot of people are going to die before we solve it, but there's a lot of people doing a lot of stuff. Like we have hardly scratched the surface. There's a lot of comfort knowing that there's so many people working on this.
And there's £10 million in it as well. So there's £10 million to anyone who can solve this problem. Right, so now we come to the part of the show which I like to call What's the Cost. Now, once again, Dave's not with us, so this game would just be for Havz. I'll see if he can try and work this out. The format's going to be exactly the same. I'm going to give him 3 items, and he has to tell me which one costs the most and which one costs the least.
Bring it on, Phil.
The total cost of the Large Hadron Collider project, first one. The overall estimated cost of the International Space Station is the second one. And the third one is the estimated cost of the construction of a Jurassic Park.
What?
Yeah.
Jurassic Park?
The theoretical estimated construction of the theoretical Jurassic Park.
Okay, you know what? I think I've read about the Jurassic Park thing, and I think that's a lot of money. Just think about it. So you'd have to invest all that money into the genetic sequencing, the actual buying an island. I mean, we're talking like, it's not even possible. So oh my god, I don't even know. Okay, so because I thought the LHC was the largest project ever internationally. I think that might be the ISS now. Oh, this is a hard one, Phil. So, okay, the ISS, that's a lot of space launches, isn't it? That's a lot of launches. All the new technology you'd have to... Then the LHC, MRIs come from that because of the technology that went into it. Okay, I think I've made my choice. For most, proposed Jurassic Park funding. In the middle, I'm going to go for the LHC. And for the least, I'm going to put ISS. Oh, this is going to be so wrong.
Yeah, you're right. It's so wrong. I'm going to take them from the cheapest to the most expensive. So at the cheapest, we've got the total cost of the Large Hadron Collider project at an estimated $4.4 billion US. Really, really quite cheap. Consider the infrastructure of the LHC. Is it something like 28 miles in diameter, like the actual thing, and all the facilities as well? It's huge, but only 4.4 billion. The estimated cost of the construction of Jurassic Park was the middle one, at an estimated 28 billion US. Now, admittedly, I did pull this one from a YouTube video where they did the rundown of the estimations of the cost for Jurassic Park. A lot of it went into real estate, but one thing they did do, which I think could probably push the actual cost up quite high, they looked at the cost of cloning dogs, basically, and just used that as the cost of creating the animals.
Come on.
Yeah, I mean, there's loads
Nowhere
more.
Near.
There's loads more research you'd have to do, and a lot of funding would have to go into that research in order to create a dinosaur. But that's what they used anyway, cloning dogs.
I'm saying that I got that one right. Just
wait until you hear the next
one.
Wait until you hear the next one. That's all I'm saying.
Oh, I should have gone this.
The most expensive one was the overall estimated cost of the ISS. So that includes running costs and everything. I said estimated cost because there are so many complicated variables with the International Space Station that the estimated cost falls within a range of 35 billion to 160 billion.
Oh my god.
Which is still higher than other 2 items, but even the lower bound is higher than the other 2 items. But still, I mean, that is a massive range.
I kind of had the logic right for that, but I just decided to do the opposite.
Yeah, you decided to make it a lot cheaper. Like, you didn't think about the cost of getting something into space and how many times they do that.
Yeah, I know.
How frequently they do it.
I was just like, yeah, they must have put loads of stuff in space. And I was like, but the LHC, that's like big and stuff. Oh, man.
I love how you said, and they may create MRI machines, and just like, I'm sure that's not that expensive compared to the ISS.
Yeah, I know, exactly. Yeah.
But yeah, I'm gonna put I'm gonna put up a link to the YouTube video for the Jurassic Park construction in the show notes,
yeah,
so people can have
it.
a look and judge for themselves whether they think they've got it right or not. So we've come to that time again. It's the end of our show. Thank you for listening. Thank you, Havz, for joining me.
Yes, thank you, Phil.
Yeah, it's been very good. A couple of quick things. We're going to be doing our first ever Skepticsnightin event. It's going to be held in Fishguard Rugby Club on the 17th of July. We're holding a pub quiz, basically, and we're going to be raising money for the Multiple Sclerosis Society and having a round of Seeing Through the Myths and a few other rounds of science-based questions and other classic pub quiz questions. So it should be a lot of fun. If you're around in the area, definitely come along.
Also, we have a lot of online profiles you can now visit. So on Facebook, we're at skepticsnightin. Twitter, we're skeptics_ni. On iTunes, you can subscribe to us and listen to us on your way to work or in the evening at skepticsnightin. And also, obviously, our website where you can see our blog and our podcast, skepticsnightin.com.
And of course, you can contact us via email at skepticsnightin@gmail.com.
Cheers, Phil. It's been fun.
Yeah, thank you very much for joining us. We've been a Skeptics Night In.